Coronavirus have a very large 30,000 bases, single-stranded positive sense RNA genome encoding for a number of open reading frames. One frame encodes the spike or S protein, a class I fusion protein that mediates attachment of the virus to the cell surface receptors. Proteolytic cleavage of the spike protein and fusion of viral and endosomal membranes triggers the release of viral RNA into the cytosol of the infected cell. As I mentioned in earlier videos, SARS coronavirus 2 is the seventh coronavirus known to infect humans. SARS coronavirus and MERS coronavirus can cause severe disease, as I have mentioned in previous videos. But HKU1, NL63, OC43, and 229E are associated with mild symptoms. In this video, I will mention what can be inferred about the origin of SARS coronavirus 2 from the available genomic data. In the midst of the current pandemic, it is reasonable to wonder about the origins of SARS coronavirus 2. On the basis of structural studies and biochemical experiments, SARS coronavirus 2 appears to be optimized for binding to the human receptor ACE2. For more details, check the earlier videos I've done. While the structural studies and the biochemical experiments suggest that SARS coronavirus 2 may bind the human ACE2 receptors with high affinity, prior computational analysis did not predict it. Thus, the high-affinity binding of SARS coronavirus 2 spike protein to human ACE2 receptors is most likely the result of natural selection on a human or human-like ACE2 receptor that permits another optimal binding solution to arise. This is strong evidence that SARS coronavirus 2 is not the product of purposeful manipulation. It is improbable that SARS coronavirus 2 emerged through laboratory manipulation of a related SARS CoV-like coronavirus. As noted above, the receptor binding domain of SARS coronavirus 2 is optimized for binding to human ACE2 receptors with an efficient solution that is different from those previously predicted. Furthermore, if genetic manipulation had been performed, one of the several reverse genetic systems available for beta coronavirus would probably have been used. However, the genetic data show that SARS coronavirus 2 is not derived from any previously used virus backbone. Therefore, it is plausible that natural selection gave origin to the SARS coronavirus 2 in an animal host before zoonotic transfer, and/or natural selection in humans following zoonotic transfer. As many early cases of COVID-19 were linked to U1 and market in U1, it is possible that an animal source was present at this location. Given the similarity of SARS coronavirus 2 to bat SARS CoV-like coronaviruses, it is likely that bats serve as reservoir hosts for this progenitor. Although the coronavirus RATG13 sample from a Rhinolophus bat is 96% identical overall to the SARS coronavirus 2, its spike protein diverges in the receptor binding domain, which suggests that it may not bind efficiently to human ACE2 receptors. All SARS coronavirus 2 genomes sequenced so far have the genomic features described above and are thus derived from a common ancestor that had them too. The presence in pangolins of a receptor binding domain very similar to that of SARS coronavirus 2 means that we can infer this was also probably in the virus that jumped to humans. Basic research involving bat SARS CoV-like coronavirus in cell culture and/or animal models have been ongoing for many years in biosafety level 2 laboratories across the world, and there are documented instances of laboratory escapes of SARS CoV virus. We must therefore examine the possibility of an inadvertent laboratory release of SARS coronavirus 2. In theory, it is possible that SARS coronavirus 2 acquired receptor binding domain mutations during adaptation to passage in cell culture, as has been observed in studies of other SARS coronavirus. The finding of SARS CoV-like coronavirus from pangolins with nearly identical receptor binding domains, however, provides a much stronger and more parsimonious explanation of how SARS-CoV-2 acquired this via recombination or mutation. From these arguments, it is more likely that SARS coronavirus 2 was naturally selected and not a purposefully engineered virus.